That's a first. A double first. You started your project?
Yeah, we're gonna meet on Sunday, I hope.
Okay. Weekend work, wow. Yeah, the ambition.
I was wondering,
Yeah, everything completely literally. The smallest level of detail, no. So the basics, what I'll add here is just what I say during the lectures. But whenever there's something in the paper, yeah, the article that directly extends it, then you might have to get it. No, I about. So what can you choose? We talked about how when you make stimulus a couple of times stronger, with bowling spots, and that is an LTI and system type thing. So you have to paper about that. Going to the table a bit about something. But in general, yeah, I don't think I'm known for making difficult. Like this. I was surprised. That was that's right. They call lectures were really interesting. And then the exam, not so much. Yeah, because I looked also at ResNet and Cornet stuff. No, yeah, that's nice. Oh, is it nice? Okay, good. I thought you were gonna say like you make me study so hard. Okay, yeah. Yeah, it's
Sharing, like, hearing, floating. Oh, yeah, it's finding water. I love this rock. Hey, oh. Water. Water. I'm just trying to check the time. I don't know if I'm all this right now. There's still.
Can anyone monitor the chat for me in the Zoom meeting? Because I'm still without a computer, so yeah, I can't look at the chat. So what? Thanks. Yeah, I appreciate that. Yeah.
I texted this. It was pretty much yeah. Do you want me to raise my hand?
Yes, please. Yes, that would be greatly appreciated. I'm going to be a subject in the study again, so I haven't been able to drink any coffee today. I don't know yet, but
Study again. Work on it.
no, the study. So with money drug studies, what they do is called a randomized controlled trial, right? So you have to have a double blind, which makes no sense for a study like psychedelics, right? So you do placebo or on drug, right? And then you can just split them up by half, or you give everybody 2 sessions, right? But double blind doesn't make any sense, of course, because you grow up in those like those. So that in this one, they actually have 3 after this problem, right? You only have 2 seconds for, and then the experiment immediately knows whether they drug the subject or not. Right? So now they have 3. It's a high dose, low dose, or placebo. And I'm sort of worried. Yeah, because I'm still in the danger zone. I'm pretty convinced that I got the high dose, but I'm not fully certain. So you might not see me again.
People participated in that the study, like realize where they started the procedure so high.
Yeah, no, it feels like that. Yeah, but now you're making me worry even more. No, no, what I maybe 2 weeks ago was more than you would normally use recreational. They might be 3 times as much. The whole reason I said this, by the way, was just so I can say, like, if I'm sort of off it, it's because I haven't been able to drink any coffee, so I'm still asleep, right? That was the main point. Yeah, they were here.
Yeah.
Okay, can everybody on Zoom hear me? You can say yes or no in the chat. Well, say no in the chat if I'm just, well, you don't know whether I'm speaking if the answer is no. Is there a yes? Good.
4 yeses.
Oh, great. Thank you so much. As you see, I'm still on my iPad, so no visuals, but you know. I received one email about a problem with a project. That was project number 1, where some dimension didn't work. So if anyone's here that is doing project 1 and is experiencing the same problem, I will fix it ASAP. If you're not experiencing any problem and you're able to run all of the code, then magic. I don't know. So just that you're aware of that. The rostering exam commission thing is still problematic. Yeah, it's getting to be quite annoying. And so I want you to know that I share that emotion if you experience it. Yeah, so next week, we'll have a bunch of classes about more either fundamental topics. So we'll do MR physics, and I won't be there, by the way, so we'll have a guest lecture by a colleague of mine. I won't be there, just so you know. And then the week after, we'll have a lecture on spectroscopy, that is to look at the sort of chemical content of the brain, which we can also now do in a functional way. This ketamine experiment is actually an example of that. So they're actually measuring the chemical content of the brain over time, right? And anything that's measured over time, we'll call that functional MRI, right? Even though it's not, well, so that's functional MRS because it's spectroscopy and it's not necessarily creating any images. But so that's what's to come. Those are, you can imagine that those are sort of segues away from what you're doing in the project and from the sort of basics of the course, right? These are sort of the cherries on top in the course in terms of extra content. And these are indeed like added things to add depth to the material and to add breadth to the material. And so I ran out of lectures, and because last year we had only half of the lectures, can you believe it? And so I decided to, we're at the end of the course more or less in terms of basic material, and so now I wanted to talk to you about what we're doing in the lab. So what we as a center, like at the Spinoza Center, what my group is doing in terms of science. So what I'm going to do is run through a couple of recent publications in the lab to show you how we are using MRI to answer our specific questions. And you'll see a bit of overlap between the projects and what we're doing here. But other things are quite different. And at the end, you'll notice that we're sort of trying to bring together different things, such as this single unit, single voxel encoding model type of thing, and functional connectivity, which I think is really interesting. This may be a bit sort of specialized in some cases, but at the same time, this is all of this is quite hot off the press, and it's stuff that we're doing ourselves. So if you have any questions, I can go in depth, right? So yeah. Yes. But again, like in terms of the, I'm not gonna, so the papers that I'm talking about aren't exam material, right? So we're gonna stay close to the surface. I'm not gonna try to drown you in stuff. Yeah. Good question. Yeah, and so for sound, I think I'll sit down, but yeah, it's kind of boring, but you know. And of course I have to, this thing doesn't work. It's annoying. So in my lab, what we're doing is we're trying to find this sort of link between AI and psychology, right? So I teach this course, for example, is both for AI students and psychology students. And it's kind of, I was trained as a biologist and physicist when I was half this age. You know, it's what we're doing is really this blend of these four different things, right? So we're using a scanner that's based on physical principles. All of the things that have to do with getting these data are really like computer sciencey types of things. We're looking at biological underpinnings of psychological phenomena. That's the idea, right? This is not a single field. This cognitive neuroscience, the way that we try to do it, is really a blend of all of these different fields. And so there's a backbone to this lecture, and that's that I'll be talking about how we try to relate the brain to neural networks, and how the neural networks that we use for example, image classification are actually modeled after the brain. So I'll talk a bit about that first, right? So if we have these convolutional neural networks, so very simple ones, right? So the ImageNet 6 years ago type things, they're really strongly inspired by how the human brain works. All of this is very trivial for you that have had courses, of course, but we'll get there. And the idea is that the visual brain consists of these, I'm pointing at something, but that's only for you guys, of course. It's a very special thing, right? You can see this, the people online can't. So at the top of the left figure, you see a monkey brain in this case, because if this were a human brain, they would be having problems. And what you have is this visual system in the primate brain. The visual information goes from V1, this region at the back, in the occipital lobe, as you learned on Wednesday, right? And then the information flows down in front, or to the front, and that's the lower part of the temporal lobe. And as it does this, the information flow, it goes from a very simple processing to more and more complex processing, right? And this idea that the convolutional neural networks are modeled after this architecture is really evidenced by the fact that these are layer cake type networks that are convolutional. So they look at a small part of the image, each unit, and they have many different layers. And the layers become smaller in terms of image extent, but the features that they are sensitive to become more and more abstract, right? So that is at the lower layers of both the convolutional neural networks and the brain, you have these simple sort of feature detectors. And as you move up this chain of processing, you get more and more complex types of processing, like you see on the right here. And if you're all the way at the top, in the brain, we don't really know what all the way at the top is, because, there's no little, that, what's that called? Men in Black, where they kill the alien and it's this little dude inside someone's face pulling some levers. That doesn't really exist in the human brain, right? So there really isn't an endpoint to where the information flows, but in a neural network that does exist, and that's the fully connected layers that you do the final classification on, right? That gives you the percentages of this is a dog, this is a cat, and so forth. Right, so at these highest levels, we know that the receptor fields in the brain and that the units in the convolutional neural network have these really, really sort of funny looking abstract type of proto face or face-like or house-like type of receptor fields. That's all not so new, right? So what this means is that these convolutional neural networks, they mimic the structure of the human brain that we know, right? But then there's a part of the human brain's architecture that we don't understand, like this part where I was talking about there is no little man inside your head. We don't really know that well, what all those things that the little man would be doing, how the brain is doing them, right? This very complex decision making, that sort of thing, is a very difficult type of problem. We don't know how that works in the human brain, right? And that also makes it harder for us to model it using AI, right? So especially for things like high-level cognition, we really have very shallow understanding of how the brain does this. And one thing that sort of highlights that you see this. So this third statement in the points list is really about how the brain does high-level cognition, and then the last one is really about how in AI we might try to mimic high-level cognition. And we really don't know how, right? This thing that I was talking about with the self-driving car at some point, right? You need a lot of world knowledge about how the world works in order to steer a car well. Yeah? Yeah, oh, that's a very good question. So people think that things like simple actions like moving your hand, or simple perceptions like telling a voice from a dog barking, that sort of thing, is low-level cognition because it's either sensation or action. Then there's like basic reinforcement learning things that you might also say is a low-level cognition, but high-level cognition is making life choices, right? Even when you're in a social conversation, trying to guesstimate what the other person means when they make an ambiguous remark. That sort of thing is incredibly complicated, right? And especially these things that have to do with social cognition or with future planning, they are thought to be high-level cognition. Yeah. So these are also the things that are very far removed from the low-level cognition. Yeah? So we know that there are brain areas that do this sort of thing, but we don't know the computations involved. Yeah? Clear? So that's the sort of setup, right? So we have a lot of knowledge, but we have a lot of knowledge only on low-level processes. And then the question becomes, okay, but how does high-level processing come about? Social cognition, for example. Yeah. So I want to point at a couple of things that might have to do with, these might be ingredients of high-level cognition, if you will. And so if we want to really make new AI, there's a few things that we need to try to do. And the same goes for if we really want to understand the brain, we want to understand how the brain does these things. And so one of the things that's really fundamentally true for all animals is that we exist in the world, right? And that means that there is a continuous feedback loop between us and the world. And when we act, that's when we engage with the world. And this is really a fundamental sort of stream that we have, right? That really drives the way that our brains work. But if you look at most AI type stuff, if it's like language recognition, like natural language processing, or this like this game thing, like even the what's that Go thing, AlphaGo, all of those are very limited, right? There's no real interaction with a complex world. It's a game, for example, right? Or it's a simple stimulus, and the choice that the AI needs to make is very simple. Yeah? So one ingredient is this interaction with the world. Now, the other is we, as individuals, have a life, right? This is also part of that first point, but we actually have a life history, right? And based on that life history, we are compelled to do certain things in certain ways and not do other things, right? And what we do by the basis of that is that we have memories, right? So we have episodic memories. That happened when I was there, sort of thing. Yeah. That's also not really something that we have very good AI for, right? There is a nice AI for navigation, but the relation between, so in our cognition, there's a strong relation between navigation and episodic memory. Like when I said, when I was there, this happened, right? When you were there, it means that there's location and time are all bunched together there. And then one thing that we do is that, we're in this world, and at the same time, we get in visual information and we get in sensory information from audition, touch. We do things that give us back feedback from the world through these senses. And when we experience the world, really have a sort of unified perception of what's going on, right? I'm not here only visually. I'm not only here sitting on my ass. I'm not here only gesticulating. All of those things are happening at the same time, and they form an integrated experience. I hope you have the same thing. Mine might break apart in a few hours. So these types of things are exactly the types of things that we don't have good AI for, right? And that we don't understand how the brain is doing them. So what I'll do is I'll focus on three papers, things that we found in my lab, where it seems like the brain is recycling this simple type of computation also for higher level types of processing. So maybe we already know more than we thought. That's more or less the sort of hopeful message that I want to try and send. So the first will be focused on the cerebellum, that you all know where it is now, right? It's at the bottom of the brain, behind the brainstem. That's usually thought to be involved in action and motor behavior. The other is involved in memory. That's a specific network in the brain that we haven't talked about much, but that I'll highlight, and the hippocampus, all having to do with memory. And then I'll talk a bit about multisensory integration, and that we actually find throughout the brain. Okay, so how are the senses interacting? So first, I want to briefly talk a bit about the parts of the brain that I'll be focusing on. What I'm doing here is I'm zooming through the sagittal, axial, and coronal slices of the brain in a very high resolution average brain. So this is, I think, 168 brains warped together and then averaged. What you see is that in the cerebral cortex, that sort of cauliflower-shaped thing at the top of the brain, this part, or this part, for those of you that can see with my mouse pointer, you see it's kind of vaguish, and that's because people differ a lot in those parts of the brain, right? And that's why we would do surface-based processing, as we've discussed at some point. But for the lower parts of the brain, so that is anything near the cerebellum is really beautiful in this sort of scan. The brainstem is really beautiful, and all of these internal structures of the brain are very well matched across subjects. So for those, it makes sense to look at data in this way. So what I'll be doing is I'll focus on the hippocampus here at the bottom. I'll focus on the cerebellum also here at the bottom. And for things in the cortex, I will use this type of visualization that we've also talked about before, right, in order for us to explain the project. But I made this little thing where you can actually see me folding out the brain, right? So this is the way that we'll be looking at that. And what you see in red here is the visual system, and in blue is things that deactivate for things that are visual. But we've gone through this before, right? So we won't talk too much about that. Now, I'll also reiterate this thing that we talked about in the encoding models lecture, right? So how do we, in terms of vision, map the cortex? And I first want to highlight that idea of that we have maps, right? We've talked about retinotopic maps, right? You all remember, sort of, right? And this idea of maps is actually a very prominent idea of how the brain is organized. We find maps all over the place, and we've talked already a bit about these different types of maps. And the idea is that if we measure these single voxels and we try to understand what each voxel is doing with this model-based approach, then we can find these preferences of each of the voxels and then show it on the surface of the brain. Yeah.
Just from chat, it's quite long. The cerebellum is so similar for different subjects. After all, motor learning happens through motor loops with its cortex. Not everyone knows how to play the same piano piece. Or was it the result of not doing surface-based, but volume-based standardization?
That's a very, very intricate question. It turns out, let me go back because it's an interesting point, right? So we are stuck with the resolution that we can manage to attain in the scanner, right? So if we have voxels, in this case, about 0.7 millimeters isotropic, which means, the same size on each direction, what you get is beautiful visualizations of the cerebral cortex and, for example, of the basal ganglia. The cerebellum also looks nice, but it turns out that if you zoom into the cerebellum, it actually has much more intricate structures that you're not even looking at well at this resolution. Yeah. So I agree, and people are starting to come around to this with the person that posed the question, that cerebellum actually looks far more intricate. It is. And actually, at the level at which the cerebellum itself is organized, which is around 200 microns, these, like, its little foldings are 200 microns instead of like 2 millimeters of the cerebral cortex, you would want to be at that resolution. It's just that you'd have to kill someone in order to be able to get the voxels that small, because you'd have to scan them for 16 hours in a row or something like that. So, and even in a living brain, people would likely move far too much. And so we sort of say, okay, we're just going to look at the somewhat more global structure of the cerebellum, right? So we can't image the fine structure. We're going to look at the medium structure. And that's actually where the cerebellum does align really well across subjects, right? So all of these minute foldings are very, very special for everybody. But this sort of white matter, gray matter contrast that you see in the cerebellum, people are actually rather similar again. So yes, we should be doing a lot better, but we still can't, especially for the cerebellum. Right. Great. So we already talked a bit about two types of maps, right? And I really want to highlight here, these maps are really what tell us how the brain works, right? They're not blobs. I mean, as a whole, they might be a blob, but we're looking at like fine scale organization inside these maps. And that really gives us a lot of knowledge about how the brain is computing things. We spoke about these semantic maps before also, right? But I want to get back to these sort of primary information processing type things. So we know, as we discussed on Wednesday, right, that the brain, in terms of its sensory organization, the touch sensation organization, or the motor organization, the low-level motor organization, has this homunculus on it, right? So it's laid out according to a body map, a very structured representation, and we call this somatotopy. And we have, of course, also retinotopy that we've talked about before. But there's topies that are much more intricate. much more abstract, like the semantic maps we talked about. And I want to highlight also these numerosity maps that we also talked about. Yeah. So these are actually, these little gradients of preference for some numerosity, like the number 2 or the number something that's 4, for example. Right. So if you zoom into a region like that, I really want to highlight this map-like structure now. Right. So I'm going to zoom into one of those regions. Look at its numerosity map. So you see that here there's a gradient from left bottom to right top, from low numbers to high numbers. Yeah. So if you know that this voxel somewhere in the middle is active and the others aren't, you know that something of 2.5 was on the screen, sort of thing. Yeah. But the nice thing is that, interesting thing here is that actually in this region, we know that there is also visual maps, and we can actually visualize those visual maps also on the same patch of cortex. So here goes, I'm going to switch to the visual map. See? And so what happens here is that you have these different maps for different modalities that overlap, and they multiplex, right? So you know now that if there's a neuron that fires here, it might be, or if you see this voxel light up, it might be that there's the number 2.5, but it also might mean that there's something on the left side of the screen, for example. Okay? And so the brain is composed of these maps, and all these maps are sort of interposed, and they multiplex. And you can imagine that this helps a lot if your brain wants to bring together different types of information processing. We think that that's really what this sort of overlapping thing is doing. We've already gone through this, right? This is the that PRF experiment. I just wanted to highlight that if we do this population receptive field mapping experiment, we see these again waves, right, on the surface of the brain, because there's a stimulus that causes these waves. Okay? And in this case, you're looking at this fixation mark, and this bar passes, and you see this wave traveling. And I just did want to reiterate this point, right, because we're doing a lot of this encoding model stuff in the lab. So what we do really is we try to fit models on the responses of every single voxel, right? So even though on the surface of the brain you have these maps and you have these waves traveling, what we're interested in is really understanding what each voxel is doing. You've seen this before, right? But in the case of an experiment like this, you see on the right top, what we're fitting is a model of what part of visual space this voxel likes. Yeah? So when we fit that, we fit these parameters, and we can explain the time course. Yeah, and this is what causes these retinotopic maps, right? This is what allows us to build up these retinotopic maps, and this is also exactly the type of structure that these convolutional neural networks are trying to mimic. Yeah, this stacked image processing type architecture. Clear? Well, it's a repetition of what you've already seen, but yeah. Great. So, and this is something that I want to really highlight in this, that I want you to take away from the course, right? The type of brain imaging that we're trying to do is really trying to step away from this is happening here sort of thing, right? This is the fear center of the brain, for example. What we want to do is just focus away from finding activation, right, but trying to find models that allow us to explain what the brain is doing. And that's a completely different type of approach. And so the game then becomes not to measure more subjects or find an activation somewhere. Right? The goal becomes to have models compete to explain what the brain is doing. Right? So you just want to find the best model for what the brain is doing. That's a completely different game. And what we decided to do was to start to take not even our own data, but use this development in the field where people are creating huge data sets. And we thought, yeah, we could now like spend a lot of time doing our own experiments and finding out specific things, but what if we can just slightly change a question and then be able to use these huge data sets that people have created. And so that's what we did. We started to look in the data set called the Human Connectome Project, where they scanned 175 or 74 subjects. Actually, it was 183, but a couple weren't so good. And there's a lot of data. So these people participated in something like 6 or 7 fMRI sessions. The anatomy that we went through just now is also from this same group. And it's just very good data. If we had to run this experiment, it would have cost us millions, and they did it much better anyway. So we decided to just start using this. And so in the things that I'll be discussing, I'll be talking about 3 different experiments that they did on this huge group of subjects. One is this retinotopic mapping that I just explained. Another is movie watching that I think is really interesting. So I think one of the Perusal papers is also about that we might have to start using movie watching type stimuli instead of resting state. But here we have both. So we're going to look at both movie watching data, the brain really doing something complex, and this resting state that we talked about also before. So the first thing, right? This action. If you ask many of the neuroscientists whose work has anything to do with action, they'll say things like, the only reason for any organism to perceive is to guide action, right? So you have these jellyfish. Here we go. You know the drill, right? So there's these jellyfish, and they have 3 different stages in their lives, right? So they are like eggs, and then they hatch, and then they swim around for a while, and then they attach themselves to the sea floor, and they become like an anemone type creature. So when they're floating around in the sea and they're swimming, they have a brain, right? They actually have a fully developed nervous system, and they hunt for prey, and they, that's their lives. But then in this next life stage, they actually settle on the sea floor. They're fixed. They don't need to act anymore, and they actually resorb their entire brain, right? Like a metamorphosis type process, and then they just sit there. So that highlights really that an animal has a brain because it needs to do things, right? I think that's really a fundamental thing, right? Our brains serve to guide actions. Like, we might think a lot about our thoughts and our perceptions, but ultimately they're there to guide actions, right? Or inaction, but that's sort of the same thing. So if you look at standard classification type AI, their actions are too simple, right? It's just saying whether something is a dog or a cat or a golden retriever or a fox terrier or whatever, right? But for organisms, things are much more complicated, right? We need to forage, we need to feed, we need to mate, we need to teach, we need to learn, all of those things. It's complicated, right? And we think that one region that really is instrumental in this is the cerebellum. Yeah, it is really evolved specifically to fine-tune action, we thought. And it turns out more and more that we're finding, like if you look at the recent literature of the last 3-4 years, it turns out that people are finding more and more in the cerebellum. So where people first thought that the cerebellum was just doing action, now we're finding all sorts of things inside the cerebellum. And I want to highlight one example of this. So what we did is we used this Human Connectome Project retinotopy data set, which is just 175 people doing exactly their experiment that I just outlined with these bars, for example. Yeah. And what we found is, and this was completely unknown, that the cerebellum actually has these beautiful retinotopic maps inside it. And I'm just gonna slowly go through what we found, which I think is pretty cool. So we didn't think that the cerebellum was doing anything visual, and it turns out that something like 30% of the cerebellum is actually organized according to visual information processing, right? So that's pretty novel. But what's interesting is it's actually very intricate. So when we first started looking at these data, we were like, hey, everything is on the wrong side, right? So we knew from our own cerebral cortex work that, you know, things that happen on the right side of the visual field, they should happen in the left hemisphere of the brain, right? And vice versa. And then we started looking at the cerebellum, and everything was the opposite way. Everything is ipsilateral instead of contralateral. The left visual field is actually worked on by the left cerebellar cortex. First, we were a bit confused, but then we went back to our neuroscience textbooks, and then everything became clear because, like we talked about on Wednesday, the cerebellum actually does everything crossed relative to the rest of the brain. Yeah? So it's really like the mirrored version of the rest of the brain. But that was just us being stupid. It turns out that when you look in detail at these retinotopic maps in the cerebellum, they're actually very interesting. And that's really also what I wanted to go into, because I think it extends this idea of these encoding models a bit further. You can actually start looking at how specifically some region is working with information. So this incredibly complex figure, I won't go into too much, but what we found was, for example, if you focus on figure D there, this is eccentricity. So that is how far does something like a visual stimulus to be from where you're looking. Yeah.
Why does the cerebellum ever get crossed? What are brain regions do?
So the Y of the cerebellum, yeah, or the Y of the uncrossing of the cerebellum, we don't know. So it's one of those evolutionary history type things there. It's completely random. We know exactly where the fibers cross. So in the pons, there's a relay nucleus that actually performs this crossing. So all the fibers from the cerebellum actually cross in the pons towards the rest of the brain. But we don't know why. It's one of those things like, if you look at the eye, for example, I'll remember that you're wanting to ask a question. I'll answer your question in a bit. Oh, just go. Yeah. Yeah, so for example, in terms of the eye, right, in terms of the retina, there is people always say, these intelligent design people, right, the religious people, they always say like, wow, everything's so complicated, you need a god, otherwise it couldn't have been designed this way. If you look at the retina, it's designed like shit, right? It's the retina actually is, it has all of the output fibers on the side where the light falls, right? So all of the machinery of processing is actually obstructing the light, and you have the photoreceptors on the other side, on the wrong side of the retina for us, right? For example, octopuses don't have this. They have better eyes than we do because their retina are in the right order, or organized in the right order, right? We don't know why this is, right? It's just some happenstance accident of evolution. Yeah. What was your question?
Yeah, you were saying something about on the lights and then about the, but I know what we're looking at.
That's a very good question. Yeah, I'm going to explain. Yeah. I already said, like, the cerebellum, the way that it's organized is too small, right, to really make sense of. Yeah. So we need these shorthands, right? And already when we're measuring even very small fMRI voxels, because we have a very good scanner and we're really proud of it, we're still doing something at a scale that's not really appropriate. Still, we have this sort of global white matter, gray matter structure of the cerebellum. And the way that we can unfold the cerebral cortex, right, by seeing where the boundary between the white and the gray matter is, and then inflating it, for example, we can also do that at the wrong scale, but we can also do it in the cerebellum. So what you can do is take the cerebellum, delineate where the edges of the white matter are, and then blow it up and cut it such that you can sort of wrap it open. So you're looking with these flattened cerebellar maps, you're looking at the back of the head, and someone has, like, if you see, like, it's normally like two fists, it's like crumpled up like this, and what you're doing is you're unfolding it like this. So that's like you're looking at the backs of two fists and they're unfolding like open hands. I don't know if that's figurative enough. Does that make sense? Does that clarify things a bit? Because it's always very hard for me to understand what I'm looking at here also.
Yeah, no, that's what I was wondering, but we don't need to look at the differences between them.
No. I'm just going to highlight one specific thing and then we have a break, okay? So, so one of these organizations of these maps, one of these like directions of organizations is how far something is removed from where you're looking, right? So eccentricity, something is far in the periphery or close to fixation. That's called eccentricity. I really need a break. And that's opposite to this angle where the boundaries of the maps we saw before. So what you see here is that something very close to your fixation is blue, and something very far removed is red. So you see that this region here is blue, meaning it has a very, very focused representation, right? All of its visual activations are for things that are very close to where you're looking. Whereas if you look at this bottom one, it likes things to be very far away. And it turns out that this is really cool because this here is a region, the center region, that blue region at the top, is a region that does eye movements. And when you're doing some experiment like this, you really need to keep focused on the center of the on the fixation mark, right? So it's the attention of trying to keep your fixation. Whereas the other regions, they're actually overlapping with, oh, I don't have that here, sorry. They're actually overlapping with the body maps that are also in the cerebellum, right? So the way that I showed these numerosity maps to be overlapping with the visual maps in the parietal lobe of the cerebral cortex, here we're finding that these regions are overlapping with like the hands and the feet. And that necessarily means that they should be looking at the periphery of the visual field. All right, so what we're seeing here is these regions where the exact way in which they like visual stimuli to appear in the visual field corresponds to what we think their function is, right? So really being able to map out in detail how these regions like things in visual sense allows us to find out things about their function. Yeah. Please.
Question, could you perhaps use the computer mouse to point at what you're talking about, but you're using it right?
Yeah, sorry, I'm using an iPad. As soon as I touch my screen, it will go to the next slide.
And then another question, do we also have cortical magnification in the cerebellum?
Yes. So this idea of cortical magnification is that you have much stronger representation of the center of the visual field and less so in the periphery. And visual regions differ in this cortical magnification, right? So this difference that I pointed out for that upper region and the lower region, that is a difference in terms of cortical magnification also, yes. Yeah. And you can tell from this cortical magnification, so what parts of the visual field a region likes best, you can tell a lot about its function. And that's so, yeah. Like if you see where the roads are in the country, you know where the economy is strongest, right? That sort of thing. Well, that's a very wide metaphor. Okay, we definitely need a break. Well, let's go back here in 10 minutes or so. Yeah.
How does the bar look like with an angle represented by the
With an angle represented by
blue color?
what that looks like.
How does the bar look like with an angle represented by the blue color?
That bar would be very close to the fixation mark, I think. In D, at least, I think we're talking about, right? Or
What's your?
yeah. I'll go to the chat and try to answer it in the break. Yeah. So see you guys back in 10 minutes. Yeah, so this is what that looks like when you when you sort of zoom through the back of the brain, you actually see these retinotopic maps in the cerebellum. And you can actually compare them with the other blobs in the midbrain that are also visual. But yeah, oh, this also shows this overlap between these visual maps and, for example, the hand maps and the feet maps. And so one of the things that we think that the cerebellum is doing with this visual processing is something like this. It's a beautiful paper that just came out yesterday in PLOS Computational Biology, where they did this. They had people wear a backpack full of computer stuff, and they did binocular eye tracking and head tracking and everything. And then they would also track all of their limbs positions at the same time. And then they would just send them up a mountain so that they would just walk up a mountain over boulders and stuff like that. Because they wanted to start looking at, okay, how does in the real world our body and our eye movements and our vision and our head movements, how do these align? Right? We don't know this, right? But if you want to build AI that navigates in the world, for example, you definitely need to know these things. So this is the type of thing that that they did. And you have to imagine that, okay, what brain areas are responsible for the type of processing that you're doing here, right? So you see on the left, you see the dots are where people are going to place their feet. And you see that they're looking at where they're going to place. I really love this movie. It's and so you can imagine that if you have a brain region that incorporates like this visual information with knowing where you're going to place your feet, well, you need to overlay vision and your feet processing, right? They need to connect somewhere. They need to multiplex. And so we think that especially those lower regions in the cerebellum that we found are visual, that they interact with feet-related processing and hand-related processing for if you're grasping things or if you're walking, right? So I think this is a nice example of how, like in the real world, our brains are having to function, right? We have to have sort of all this functionality needs to overlap. Yeah. Okay, so here you see equally sped up versions of on the right, a retinotopic mapping experiment and a documentary on the left. And so I don't know which one of you, each of you finds more interesting, but I definitely appreciate the left one. Yeah. And one of the reasons for that is that on the left, that's what we evolved to look at, right? Not movies, to be clear, but Hollywood isn't that important. But this is showing us things that our brains are made to understand, right? Actually understanding what's going on in a movie is very, very similar to what we're doing in everyday life, right? And in many cases, a movie is much more potent even than what we're seeing in everyday life. For you guys, definitely, if you're looking at me or at the movie is much more interesting. Right. So the main point here is that if we juxtapose these two things, if we contrast them with one another, the mapping experiments are boring. They're very functional, right? But they allow us to look at what the brain is doing. That's all great, but they don't allow the brain to do what it usually does and what it does best, right? We use vision to understand the world around us, and there's nothing to understand in a dim-witted bar that's crossing the screen. Right? So we have a big problem if we're trying to say, okay, we're just going to do that right experiment, and then we're going to pretend that like we're understanding the brain processes that are used to doing what's on the left. That makes very little sense, right? So in some way, we have to try to find what's going on in our brains when people are looking at what's happening on the left. And so this is where this second paper comes in, because we know a bit about the architecture of the visual system from monkey tracer studies. So that's not functional, it's just those tracer studies that trace how the axons run in the visual system of the monkey. and there people found that that all of the roads in the visual system seem to lead towards the hippocampus. And the hippocampus is not considered to be part of the visual system. It's a memory related region. It's not supposed to be in the visual system, but these monkey findings show that it's really the pinnacle of the visual system. So that's a really weird conundrum, that this hippocampus, we don't never find it in these bar experiments, right? Hardly ever does it pop up in a bar experiment like that. But it is part of the visual system because there's anatomical connections. When you think about it, it sort of makes sense. So the hippocampus is this memory related region, right? And what it does is it really tries to integrate over larger portions of time and like events, things like that. That's what hippocampus does. So it constructs things like event boundaries and tries to function as like a memory lookup table, right? This episodic memory that we talked about just now. And so we started thinking about this, and then we came to the conclusion that, well, if hippocampus is part of the visual system in this anatomical way, and we're not finding it because of our simple experiments, maybe that's just because hippocampus is involved in exactly that type of visual processing that you need during the movie, but you don't actually require during the simple mapping experiment. It's this visual understanding. What am I looking at? Yeah, because that's what has all this world knowledge in it, right? What am I looking at? Ah, this is this and this. You know that a hand is likely to grasp things or drop them, right? So things make sense, but you need a hippocampus for this type of understanding. Yeah.
But the understanding also corresponds to recognizing objects.
Yeah. So yeah. So hippocampus is important in that sort of thing. But recognition is a bit of a vague concept in this way, because just classifying whether something is a cat or a dog is also like a recognition type thing, right? And that is done by the ventral visual system. But people have looked at, so for example, humans have a huge memory for scenes, right? So if I show you 100,000 pictures, like in a row, 1 second each, and a week later I ask you, have you seen this before, right? You're actually pretty high, right? It means that your brain is able to recognize all of these separate instances. And these are so unique that people think that's also where the hippocampus plays a role. The hippocampus connects to that whole ventral visual system to lay down these memories. And that's quite different from, is this a cat or a dog, right? That's and so just one thing that I don't know if you've realized this, but if your visual system is stuck to the, let me go back to Homer because he's awesome. But so the visual system, as we map it with these bars, is actually stuck on the retina, right? And what that means is when you move your eyes, all of the inputs change, right? So the entire visual system changes its inputs whenever you move your eyes. You move your eyes around all the time. And this means that what the visual system is doing is actually very, very confusing. This what we call invariance to eye position, this sort of stuck to the retina, that actually changes as we move up the visual hierarchy. So in terms of what the hippocampus is doing, it isn't actually encoding things in necessarily visual coordinates, but it's much more known for this allocentric reference frame, that the cells there encode where an animal is in a surrounding, and not just where a stimulus is on the retina. And so it does much more abstract types of processing. And the question really is, how does the brain construct this type of abstract processing? So we have all these sensory fundamentals. How does the brain make sense of them and how does the brain start to understand them in an abstract way? So one of the things that we decided to do was to try to start looking at the hippocampus. And we were a bit baffled by this, because normally when you do a PRF experiment, like this population receptive field experiment, you need people to look in the center of the screen, because you're looking at this retinotopic thing. If people move their eyes, it shifts the stimulus, and then you have a hard time figuring out what's going on. However, when people are watching a movie, no luck, right? People are just going to look around, and everything changes all the time. So it becomes really, really hard to figure this out. And it turns out that someone a couple of years earlier had devised this method called connective field modeling. It's very similar to this population receptive field modeling. But we realized that you can start to apply this during movie watching. So let me try to explain the catch. If anything is unclear, ask. Yeah. So in this population receptive field mapping, what you try to do is you take a voxel's response and you fit a model in visual field coordinates. Yeah. So that's like where on the screen does this voxel like things to be. That's what that model fitting does. But now we really don't have a good model of what's on the screen, because people are moving their eyes a lot, and it's a very complex movie, incredibly hard. But with this connective field modeling, maybe we can solve this. So this is sort of a blend between this population receptive field mapping and the functional connectivity things that we talked about in the connectivity lecture. So what you can do is not set up a model in visual space, but set up a model in brain space. That is to say, if you have a map of the visual field in this V1, this region of the visual brain, I can make a model that's a Gaussian on the surface of V1. So I can place this Gaussian everywhere, just move it around, right? The way that I can move the PRF model around, I can now move this connective field model around, not on the screen, but in the brain. And I can also change the size of the connective field model. Okay? So what I want you to realize is that we're doing more or less exactly the same. Yeah. Each of these is a model, like in the PRF thing, you have these Gaussians in the visual field, that's a model. These are also models. Now in the PRF case, in the population receptive field case, we have a movie of what's on the screen, so that we know that when this PRF activates. For this, we can just use the brain activations in V1. So we don't have a movie of the screen, we have a movie of what's happening in V1. So what does that mean? We can take this connected field model and just multiply it with this ongoing time courses in V1. And what comes out is one single model time course. The way that a population receptive field model also combined with a movie gives us one single model time course. And those we can then find, we can just fit them on the brain. Right? And what this allows us to do is find this structure of V1. It's a beautiful retinotopic map. We can actually try to project it into the rest of the brain. Well, so what this means is that it seems to be stuck a bit. Yes. So what we can do is it doesn't matter where people are looking anymore. We can just take this movie's impression on V1 and see how the rest of the brain correlates with V1. So more or less what we do is we project V1's retinotopic map into the rest of the brain. And it doesn't matter whether people are making eye movements anymore, right? Because the eye movements, they would happen, they would shift things on the screen, but they don't shift things in V1. V1, it doesn't matter whether the eyes moved or not. There's just a pattern inside V1. It doesn't matter whether people are making eye movements. We actually, if all of the brain processes that are engaged in things like eye movements, we can actually capture them with this. And the beautiful thing is also, because we don't care anymore what was on the screen, we can actually directly compare between different conditions. We can compare between a movie watching, and we can compare between this resting state. So we can actually we stop caring what was on the screen. Yeah.
Can you look at the whole area of V1?
Yeah.
All at the same time.
Yeah, so specifically what you might get, and I think I'll explain that in a bit. So let me go back a bit. It's very slow, something's wrong. Here we go. Yeah, let me explain this by the I'm a bit anxious to so if you have this, so let's say you have a voxel somewhere here in V2. It has its time course, it has the time course of its signals, right? And it has that time course by virtue of the fact that it's also on a record-topic map, because it matters where things are. And so that means that if this voxel in V2 has a specific sort of time course, it's going to share that time course with things in V1, but it's going to share that time course with only those parts of V1 that actually have the same record-topic location that they like. So, so this model allows us to test that. So for every part of V2 or V3 or anywhere else in the brain, we can now check what specific portion of V1 does this voxel correspond to best. Now, that allows us to say, okay, when this voxel in V3 likes this portion in V1, then that means that it likes this portion in the visual field. So I can actually project that retinal-topic map of V1 into the rest of the brain. Yeah. Just by virtue of the fact that the time courses are dependent on the fact that they all like the same part of visual space. Yeah. So that's that retinal-topic map of V1. And with this functional connectivity measures, based on this model, we can now project them into the rest of the brain. And this is when it gets stuck. So it turns out that when you do this during movie watching, so this is inflated versions of the brain and the flattened version in the middle. Most of the brain has this retinotopic structure. So one of the questions in the break was, but if it's all about blending, for example, action or higher cognition with vision, then everything should be visual. I agree. And that's actually more or less what we're finding. Right. So what you see here is how strongly or how well this model explains the signals throughout the brain. And anything that's even remotely purple is already significant because we have so many subjects in this huge data set. You definitely see that the traditional visual system is most visual. It's completely yellow here, right? So this is the strongest link to V1 is what these regions have. But you see also in like areas like frontal cortex, you see a lot of visual processing happening. And the thing is, this mostly happens by virtue of the fact that people are engaged in movie watching. If you do the same analysis for this retinotopic mapping where people are forced to fixate and there's just this simple bar passing on the screen, you don't find this, right? So this is where we think that this supports this high-level cognition that allows us to understand the movie, and that much of this processing is actually in this visual framework. So for lower-level cortex, you can actually recreate the retinotopic maps from V1 in the rest of the brain. So you actually see these red lines between V3, or so outside of V3, or the green line between V2 and V3. You can really find the structure of the visual system throughout. Yeah. Yes. So people are doing it. People are doing a lot of like storytelling type things in blind people. I think that the general finding for blind people is when you do an experiment like this, their visual system starts to become auditory, right? So they actually, you can decode what sounds people are listening to from visual cortex if they're blind, but not if they're sighted. Yeah. So what in general we think is that when people, especially when they're congenitally blind, a lot of the brain that we tend to devote usually to vision, right, becomes repurposed and starts to become adopted by sense of touch and audition, for example. Yeah, yeah, definitely.
I guess that's why they have usually a better hearing, better sense of touch.
Yeah, yeah. So you have these blind people that can do echolocation and things like that. I mean, we could learn it, but opening your eyes is much easier. Yeah.
So you have to look at the V1 or
Yeah. So this recruitment of vision by auditory information processing in blind people is strongest in those regions that have some auditory information processing anyway, right? So in high level visual system, these things integrate already. But in blind people, even V1 starts to activate for auditory things. Yeah.
Question from the chat. Figures irrespective to condition show similar activation.
Yeah, that's a good question. So just to clarify, again, what we're looking at here is not like activation, but it's really the map-like structure, right? So we're looking at the what part of the visual field every vertex or voxel on the surface likes. And what this figure shows is that, for example, if we look at V3, right, it goes from, on the left, it goes from purple to light blue. And it's supposed to, because you find that in retinotopic mapping, right? It goes from purple to light blue on the top. There, I can point at it for you guys, like here, right? It goes from purple to light blue there. And you see exactly the same thing in movie watching and the resting state. What that means is that we are able to sort of retrieve, to find the organization of the visual system, even if we don't do a retinotopic mapping experiment, right? Even if people are just doing a fixation and in the dark, you can find from the ongoing activations in the visual system, you can find that structure. And the same goes for movie watching, but it becomes much stronger. You can see that these lines are much stronger in these cases than they are here. In the resting state, everything blurs out of it. I hope that answers the question. So you're not necessarily looking at activation here, you're looking at really the map-like structure of the visual system that we were able to reconstruct using this connected field mapping. So this is taking a while. Yeah, so quickly, there's one other set of regions in the brain that connect very strongly to the hippocampus, right? You remember that this whole story was still about the hippocampus. There's one specific region, set of regions in the brain that connect really strongly to the hippocampus, and they're called the default mode network of the brain. And they have to do with like autobiographical memory and that sort of thing. Is that like someone using a power tool? Wow. Yeah, well, let's hope they stop. And so we know that these regions in the brain that are blue in the left figure, or in the right top figure, these regions are blue. That's because they deactivate whenever something salient happens. And they activate for specific social and stuff categories. Like these are the semantic maps. So these regions in the brain, they like social types of information processing, and they deactivate for sensory stimulation. And that's what you see here. And these regions are connected to the hippocampus. Yeah, this is not really working, is it?
People are getting stressed again.
Yeah, I'll have a look at whether they're in this floor. I'll be right back. Do you think it's coming from this floor or from the floor? Well, it's not like it's coming from there, right here, right?
Yeah.
Did it reappear or not?
No.
Really, the poltergeist again, yeah. Okay, let's hope for the best. Okay, so we have. I'm getting stressed now. No, so we have these regions in the brain that we think are involved in this high-level cognition, right? This social information processing. Yet they also do things in when people are focused on the outside world in terms of perception. So we're interested in exactly these regions. So we wanted to look at whether this what we call retinotopic connectivity, right? So the fact that we can use this layout of V1 to explain responses in the rest of the brain, and then see those brain regions in the rest of the brain have visual structure. We wanted to understand how this depends on this cognitive state. I just pointed out a difference between the movie watching and the resting state, and we wanted to go further in this. So that's what I'll be talking about here after this question.
Yeah. So one more question about that V1 structuring is like the response of V1 to another area bigger when watching a movie or smaller when resting? Is that like the main point?
Yeah, so that's what I'm going to show you after right now, right? So how strongly is something visual, right? And how does that depend on what you're doing? Yeah. And so that question posed in another way is this retinotopic connectivity dependent on cognitive state? Yeah, on the experiment that you're performing. Yeah, because you can imagine that some regions in the brain are going to be more visual if you're doing one thing and less visual if you're doing another, and vice versa. Yeah. So yeah. So what we can do is exactly answer this question by looking at this resting state versus movie watching, right? This is a very good contrast because in one case, what you're experiencing is driven by the movie, right? And it's a very rich stimulus, completely driven by the movie. And in the other case, you're just lying there thinking of your grandmother and your shopping list and all these things, right? So it's all of the brain activation is generated by your brain itself, right? And that's why people find resting state interesting. But the contrast between the two is really interesting, especially if you have this measure for how visual a brain region is, which we do now. Okay? So what the upcoming figure will show is if something is red, it likes to becomes more visual during the resting state, and if it's more blue, it becomes more visual during movie watching. And what you see here is that you see all of this blue stuff at the bottom of the brain and towards the auditory cortex. So where you see STS on the right hemisphere, on top of that STS is where auditory cortex is. In the left hemisphere, a bit left of where it says STS is where the auditory cortex is. And of course, this is a movie watching experiment. So you actually have auditory inputs too. People are actually listening to like monologues in Ocean's 12, for example. There's a 2-minute excerpt from Ocean's 12 that people are just watching in the scanner. And so what we think that these two types of blue patches are at the bottom is that, of course, when you're looking at things that are interesting in a movie, you're recognizing things, right? So you're using vision to drive all of these image recognition things. And we know that that's in the temporal lobe, and that's the lowest part of that. So that's image recognition happening. And we think that stream outside of that, the STS stream, is really this whole comprehension of what's happening in terms of social, like people are having a conversation and you're looking at them, you're going to have to integrate what you're seeing in terms of their lip movement and what you're hearing in terms of their voice, right? So all of that type of processing we think happens in those blue regions. And you would never get that, but you would never be able to analyze that if people are just fixating at the center of the screen, looking at a bar passing by, because it doesn't talk, right? I hope. So that's for the blue part. What you also see is there's a lot of yellow, and that means that these regions really don't care whether you're watching a stimulus coming in from the outside or whether you're thinking about something. They actually have more or less the same connections to V1. They're always visual. Yeah, and this is also already quite a lot more than we would normally find in an experiment. And then there's these red regions. These red regions are exactly the regions that were red in that previous figure, where they're for social cognition. And it turns out that they sync up with the visual system much more when you're left to your own devices. So your endogenous thought seems to be linked with vision in these regions. Yeah. And what we think that means is that when you're just, closing your eyes or looking at the center of a dark screen, these regions connect with V1 because it's your imagination, right? When you're thinking about just anything, you're probably recruiting vision for this, right? Because you're having all these mental images pass by. We think that it's exactly these regions that connect up to vision to perform that endogenous sort of mental imagery that you experience when you're just left to your own devices. Yeah, that's what I just said. This isn't important. But what's really cool is I've tried to give you an idea of how important it is that we can actually see how something is representing the visual field, because it tells us something about what its function may be. And we can actually start doing that, but then dependent on whether something is, depending on cognitive state. Yeah, so we can check, does this always have the same visual map, this region? Or is the visual map that it has, is it flexible? And does it depend on what you're doing in your mind, right? Whether you're watching a movie or whether you're left to your own devices in the resting state. Yeah. And so one of those things that we can look at is eccentricity. So how focused is it on the center of the visual field? And we can look at, for example, this is one visual region, and we can, here you see that in the resting state, the visual field representation becomes much more wide, right? So you see that the eccentricity of the connected fields becomes higher in the resting state than it does during movie watching. Right? So whenever you see a scatter plot like this, the dots are above the unity line. You always plot a unity line. Most of the dots are above the unity line. You have this increase for the y-axis, right? So larger values for the resting state. So what this means is when you're watching a movie, these connective fields, these visual representations, when you're watching a movie, they all like the fovea more, so more the center of the visual field. And when you're left to your own devices, they sort of diffuse out. They span the visual field more. And this is pretty cool, right? Because it means that with this type of analysis, we can start to look at, the sort of warping that your brain's visual system performs in different mental operations. So we can look at that sort of thing. We can also look at how, this is going too fast. Well, we can also look at the size of these. So the size of the patch in V1, right? So is this communicating to a large portion of V1 or a small portion of V1? Is the connectivity between two regions, is it very precise, or does it become more broad? And here it turns out that during movie watching, things become more broad. The spatial degree of integration becomes larger. Whereas for resting state, it's mostly fixed. And we can also look at which hemisphere it connects to preferentially. Yeah.
Thinking or resting state, we also have mental images in V1 then?
Yeah, that's what we think. Yeah. So yeah. Like you have, when you have a stimulus, it appears in V1, of course, and then it gets transferred to all of these other regions. You have this concerted activation throughout the visual system, right? And if you imagine something, you have a very similar concerted activation throughout the visual system. but it's driven by endogenous processes and not by an ongoing stimulus. But you can imagine that this concerted activation throughout different visual regions is different depending on whether you're endogenously activating it or whether it's driven by a stimulus. And that's what we're looking at here. Yep. So again, I'm not going to quiz you on all of these graphs, right? I just want to give you an impression of how strong this idea is of trying to understand what's happening in every voxel, right? And trying to relate it to what's happening in a sensory region. So for this visual system, we have very strong correlations. So it doesn't really depend that much on what the subject is doing. You have this relatively fixed sort of structure of the visual system, and it really doesn't depend that much on what you're doing. I won't go into this specifically. So remember, I was talking about the hippocampus, right? That's why I wanted to be looking at this. This is a region that is supposed to play this pivotal role in understanding what we're looking at. And so we wanted to start having a look at that. So I'm going back and forth between throughout the subcortex. And you see definitely, like, for example, in the cerebellum, you see these very blue regions. That's because you're moving your eyes during a movie watching, right? And you have this action that you're performing during movie watching. Similarly, we see a whole bunch of regions in the brainstem and things like that. We know that these exist, but we also see that the hippocampus activates. And I'll zoom into the hippocampus now. So what I'm doing is I'm zooming into this patch that you see at the bottom. And here we're looking at the hippocampus. It travels up and down, and you see these activations. So this is what this is showing is that the hippocampus is actually synced in this very specific way to V1. And in most cases, you see that it's yellow or red, or in the front, it's more bluish. It sort of depends also on the cognitive situation that people are in, how strong this syncing is. So we can actually look at the different subparts of the hippocampus. They're supposed to have different roles. And this separation of function in the hippocampus, the separation of the blue and the red regions, in that certain regions of the hippocampus like to be visual in the resting state, and others like to be visual in the movie watching. That actually corresponds completely with what we know that these different regions do. So we know that these CA13 and dentate gyrus regions of the hippocampus, they are the input regions to the hippocampus. And so you'd expect that they are more interested in being visual during the movie watching. And that's definitely also what we see. So this really aligns very well with all of the different things that we already knew from things like mouse electrophysiology, things like that. And we can do the same thing that I showed you for this visual system region, but now do it for hippocampus. So you can look at how strongly the visual representations of a region are focused on the center of the visual field. And we see that it's much more eccentric. It's spread out more during movie watching. Is that what I'm saying? Yeah, then during resting state. So it's much more, so during resting state, these regions become much more focused on the center of the visual field. The same goes for the size. They become smaller when you, so these regions become, the connective fields become smaller during a resting state relative to movie watching. Now the hemisphere is not too important. But it does mean so, though, is that you can definitely draw a correlation line between these clouds, especially the middle one and the left one. What that means is that regardless of what people are doing, whether they're thinking of their grandmothers in the dark or watching a movie, you have a very stable visual representation in the hippocampus. So people had never seen that the hippocampus was visual before. And what we're seeing here is that it's very stable, always visual, but there are slight differences between how the hippocampus links up with the visual system. So we can actually, so this prompted us to go back. And then we went back and looked at the retinotopic mapping experiment, because we'd already, we found that the hippocampus was visual during the movie watching and during the resting state. And then we thought, hey, but maybe if we look at this huge data set and we start doing a very detailed analysis on the hippocampus in the retinotopic mapping experiment, maybe we can then already also find this visual organization. It turns out that we can, and that the hippocampus actually performs very similarly to a standard visual region when you start doing PRF modeling on it. So, so we've really seemingly discovered that this that the hippocampus is visual by looking at what happens in every single voxel separately and trying to understand it in terms of how that voxel relates to the sensory stimulus, the sensory brain areas, yeah, to V1. So that's more or less what I wanted to show you. I want to give you a brief outlook because the things I've shown you up until now are they're only visual, right? I've spoken a bit about multisensory things in that you know, you need to bring together sound and vision, that sort of thing. But in principle, this type of modeling that we're doing, this connected field stuff, you can do from any region. And what we think is happening is what could be really interesting is this interaction between action and vision. We're now looking at whether we can understand that by doing this connectivity modeling from not only V1, but also from the sensory motor regions. And it turns out we're finding really beautiful things. So we're really looking at how, for example, in this case, auditory and visual system interact. So we're finding new regions that are auditory in the frontal cortex, very much biased towards language processing. And people didn't know that these are necessarily auditory. So it seems like we're sort of stumbling onto a way of parceling out this map-like information processing in the brain. And so this is what I wanted to show you as a final thing. So we're now doing this same type of analysis from vision, audition, and sensation at the same time. This is also this rotating brain that's the icon for the course in Canvas is actually this analysis. And what you then start to see is that a lot of these regions that we thought we showed are visual, right? In that finding that I just outlined. These aren't actually just visual, they're also somatosensory, right? So if you look at one of those regions, it's not completely blue, it's actually somewhere between blue and green. And what this means is that we can understand what's happening there, not just in terms of visual coordinates, but also in terms of sensory coordinates. So for example, we're finding that in these portions of the visual system, things are very much related also to hands or to faces because of there are specific connections between the hand representation here and the face representation there. So we think that we're starting to get a handle of this type of organization in the brain by virtue of this type of analysis. So I hope this was a nice sort of tidbit of showing you where things are going in the lab. Yeah. I understand if things went a bit fast once in a while. If you have any questions, ask them now or over email. I want to highlight also or emphasize that I'm not going to ask details about this lecture in the exam, right? But this is really to show you, like, knowing the basics that you now have, where can you start to take this, right? So I hope it gave you an idea of what the cutting edge is in terms of what we can start to do. Yeah.
The question related to active preparation.
Uh-huh.
Free time left in one of the lectures to run through questions on the practice exam that's on campus.
Yes, yes. I forgot, did I upload the answered exam? Because I made an answered exam. But I don't know if I managed to upload it before my laptop crashed.
Is it-
No? Yeah, it should be under files. No, it's not there? Okay, then it's not there. Okay, so then I'll upload it this weekend. Yeah.